the panel review we have later on today. So these are my disclosures, and today I'd like to talk about Kabuki Syndrome associated immune deficiency. Depicted here is really one of the workhorses of the humoral immune system, the plasma cell, and this is a very classic picture that all medical students have to learn because of the clock-like face of the nucleus and all this wiggly stuff here, which is the machinery that makes the antibodies. And antibodies are proteins that permeate all the humoral tissues, all the bodily fluids of the body, and protect you against infections. And that's the particular arm of the immune system that I'm going to talk about today that we find is particularly affected in Kabuki syndrome. So briefly, we're just going to I'm going to run through what the humoral immune system is. I'm going to talk about KSAID, and then I'm going to talk about my very productive collaboration with Dr. Bjornson's lab using the KMT2D beta GEO mouse, the Kabuki mouse, and how we've been able to understand in greater depth the particular cell types and mechanisms that are affected in Kabuki syndrome and lead to this problem with making antibodies. So I think everyone here is fairly familiar with Kabuki syndrome now after the excellent talks given earlier today. But just briefly, I wanted to point out that the immune globulin deficiency and autoimmunity are features that are oftentimes underappreciated, but can significantly influence a patient's course, especially if they end up having lots of pneumonias, infections, or autoimmunity that can lead to significant complications. So as was discussed before, there are two different genes that are affected with Kabuki syndrome. They both are involved in opening and closing the chromatin, which are the spiral loops of DNA around these little spools, and the spools have tails, which the two different genes associated with Kabuki syndrome regulate and open and close. So I'm just going to go ahead to the immune system stuff, though, since we've spent some time talking about the particular genes and their mechanisms of how they regulate gene regulation. So the question I frequently get is the most important one, which is immune deficiency part of type 1 Kabuki syndrome? And my talk will be focused on type 1 today because that's where we have the most data on this particular aspect of the syndrome. And so yes, almost all patients with Kabuki syndrome, type 1 Kabuki syndrome, have IgA deficiency. This is actually the most common immune deficiency found in humans, with about 1 in 200 patients, just non-Kabuki people, having low levels of IgA. But in addition to having low levels of IgA, about 40% of Kabuki patients will also have lower levels of two other important immunoglobulin types, IgG and IgM, and the majority of them will have some abnormality within their B cell compartment. And there are lots of different types of B cells that go through a complicated differentiation pathway, and I'm going to dive into some of those details today. But to help you better understand and put this all into context, I want to briefly talk about this humoral immune system. So this is the part of the immune system that makes these Y-shaped proteins called antibodies. These are exquisitely specific. They've been described as kind of a lock and key mechanism, where a particular antibody is very, very precise in targeting a particular three-dimensional shape at the molecular level. So when you get vaccinated against tetanus, when you get vaccinated against anything else, or you get an infection, your humoral immune system responds by making antibodies that fit a very particular three-dimensional shape called an epitope on a particular protein or another biomolecule from that pathogen. And there are lots of different flavors of antibodies. The ones we're going to spend a lot of time talking about today are the IgA antibodies, which are the ones where there are two of them stuck together in the secreted form. There's also a monomer form of it, or a single Y-shaped protein that's very prominent in the blood. So the IgA is at high levels in the fluids in the lungs, in the intestinal tract, as well as at present in the blood. IgG is at very high levels in the blood, and it helps prevent systemic infections. And some of these other forms are not entirely relevant to the conversation today. I will point out IgM, which is where there are 5 of these proteins all bound together. This is the earliest form of an antibody, and it's kind of the starting point. And I wanted to point that one out because we do see some differences in that form of antibodies in one of our mouse models. So where do the antibodies come from? Antibodies come from B cells. So these B cells interact with a pathogen, or it can be bacteria, it can be a virus, it can be actually in pathogenic states, it can be a part of your own body, and you can make an antibody targeting yourself. That's autoimmunity. And then frequently the B cells will talk to another type of immune cell called a T cell. And then after that, they get selected if they are making something that's very specific, and it goes through a couple of checkpoints. You end up with memory B cells, which allow you to be resistant to a particular pathogen many years into the future. And they also make those workhorse proteins, the plasma cells, which are pumping out huge amounts of these antibodies that permeate all your tissues and all your mucosal surfaces. And then finally, these cells are in different compartments throughout the body. So you're all probably familiar with arteries and veins. This is how most cells get around the body, but there's a third form of vessel called lymphatic vessels that connect these secondary lymphoid organs, which not everyone are familiar with, but the B cells traffic around and move around the body in these lymphatic vessels, as well as in the blood. And then they also come from the bone marrow. That's where new B cells come from, are born and go out into the blood. And then also it's the home for many plasma cells that are part of your long-term immune memory. In addition, there are special spots throughout the body. You can see all those little scattered dots on this diagram. All throughout your body you have little lymph nodes, which are these places where B cells and T cells get together and kind of talk about whether they want to make an antibody against something. So your body is constantly awash in foreign antigens, and the immune system has to make a decision whether or not they make an antibody targeting that the foreign thing you've encountered, or whether they should ignore it. And this is particularly important in the other part of my work, which is looking at allergy. In allergy, you accidentally make an antibody against something that you shouldn't make an antibody against, like tree pollen. Tree pollen is not interested in infecting you, although it can make many of us miserable this time of year. Likewise, food allergy. Peanut is not trying to kill you, but in people who are peanut allergic, they make an antibody inappropriately to it. In Kabuki Syndrome, these patients are just not very good at making antibodies, period. So they generally are not allergic, and they generally are not so good at making IgA. And then there's a subset of patients that are really bad at making all antibodies. And those are the ones that need a special attention and require IVIG therapy. So finally, I wanted to mention there's a special type of lymph node that lives in the intestine called the Peyer's patch. And this is particularly important for making IgA. So now this gets to some of the work that I've done in a cohort of patients I've been working with at Cincinnati Children's that have Kabuki Syndrome for several years. And this is some data that was published several years ago. We've since expanded our cohort and have been able to validate these findings. But this is a graph of serum IgA. This is the antibody that is found in the blood, but also is very important for coating all the mucosal surfaces, the lungs, the GI tract, and it helps protect you from infection at those spots. And the gray field here represents the normal range of IgA in patients as they get older. And you can see it starts off quite low, but then as people get older, they make more and more IgA as they've encountered more pathogens in the environment. And you can see the vast majority of Kabuki patients fall below the lower limit of normal for making IgA. They basically make the level that an infant makes, and they never expand it. Toddlers should dramatically expand the amount of IgA they have in their blood around 2 to 3 years of age, and Kabuki patients, for the most part, don't do that. In addition, some patients, about a third of patients, will also have problems making other forms of antibody like IgM and IgG. These are patients, especially that have low IgG, that benefit from IgG supplementation through either subcutaneous therapy or via the IV form. When we look at the B cells in these patients, we find that many Kabuki patients will have low levels of total B cells in the peripheral blood, and in addition they have lower levels of the memory B cells, which are marked by a marker on the surface called CD27. You can see this group of patients that are all kind of living at the lower limit of normal or below for the memory B cells. And then there's another special form that make IgG and IgA called class-switch memory B cells, and the patients tend to have lower levels of those cells. And in addition, we see that in older patients in their 20s and 30s, they begin to accumulate types of B cells called CD21 low B cells. And these are B cells that are dysfunctional, they're associated with other forms of primary immune deficiency, and just indicate that the B cells aren't acting properly. Most B cells should delete themselves over time if they don't become a memory cell, whereas these cells start to accumulate in the blood of people who have immune deficiency. And it's a marker for common variable immune deficiency, which is one of the most common forms of humoral immune deficiency we see in patients across the board, not just Kabuki syndrome. So when we take B cells out from a patient that has Kabuki syndrome and we try to nudge them using a number of different activation factors to become a memory B cell or a plasmablast, you can see in this quadrant right here, I'm not sure if my pointer is showing, but if you look at the quadrant on the 7-day one between IgG and CD38, in the controlled patient, about 40% of those cells will differentiate, whereas in Kabuki patients, it's dramatically lower, around 8%. So it appears that the B cells in Kabuki syndrome are just not quite as efficient at becoming the memory B cells and the plasma cells. In addition, we know over time, that at mucosal sites as well as in the blood, the IgA level goes up. I showed you that in the blood, IgA goes up with age. We also know this is true for the saliva, and that's a site in the mouth and in the GI tract where the IgA level has been shown to increase over time. And we're in the process of trying to confirm if that's the same is true in Kabuki syndrome. Is it not only in the blood that the IgA level is low, but also in the mucosal tissues and the saliva? And our preliminary data shows that in comparison to the control subjects, which are represented in green, that the Kabuki patients they don't seem to expand the amount of IgA in their saliva with age like their normal colleagues would, or people who don't have Kabuki syndrome would. And so we're in the process of continuing that study to try to validate that as a biomarker and a potential thing we could monitor during a therapeutic trial to see if IgA goes up in the saliva when people are on certain therapies, experimental therapies that might be helpful in Kabuki syndrome. So for anyone who's interested, we are collecting saliva both from patients that have Kabuki syndrome and any sort of siblings, any children or siblings that might be along for the ride today. We would also be excited to collect saliva for them as normal controls to compare with. So our results from the human studies have demonstrated that there's a population of B cells. You can see here in this diagram that B cells start off in the bone marrow, they go out into the blood, they eventually go to lymphoid tissues like the spleen and the lymph nodes, encounter T cells, and ultimately they become this population I've highlighted here called memory cells. In addition, there are a bunch of plasma cells that come out of this process that are kind of the workhorses that just pump out tons and tons of antibody. But we really wanted to go more in depth than what we can do with peripheral blood. And so that's where the mouse model that Dr. Bjornson has developed has allowed us to go really in depth and try to understand all the different areas where the B cells might have problems trafficking from one compartment to the next, and allowed us to really try to understand the precise details of how these problems develop. So this is the same mouse system that's been described in detail thus far, and we found that the spleen, an organ that's very critical for B cell development and differentiation, is enlarged in Kabuki syndrome Kabuki mice rather, and we see the same thing in about 60% of our adult Kabuki subjects when we check by ultrasound or by CT. This is a very common finding in patients of all sorts that have a primary immune deficiency. So this is not specific for Kabuki syndrome, but it definitely is a general sign we see in patients that have problems with their B cells. So that's a part of the mouse model that recapitulates what we see in the humans. In addition, the IgA level is quite low in the Kabuki mice, just like what we see in the humans. One slight difference was we saw an elevation of IgM in the mouse model, which we don't typically see in the humans. We also, in this particular mouse model, at baseline, we don't see any difference in any of the different types of IgG antibodies. But what we did find is across developmental time, from the very earliest time points when the mice are quite young all the way out to a year and a half, which is super old for a mouse, they continue to have low levels of IgA. So it appears to be a problem that starts very early and persists throughout life. So to better try to understand how these mice respond to a challenge of their immune system where they're supposed to make IgA against a particular target, we utilize the ova cholera toxin model. And this is where you take a protein called ova, which is one of the major proteins found in egg, and you combine it with another protein called cholera toxin, which pushes the B cells to become IgA producing memory cells and plasma cells. So we vaccinated the mice over the course of several weeks, and then looked at their ability to make ova-specific IgA, and it was significantly diminished in the Kabuki animals in comparison to the normal animals. And so this suggests that it's not just a non-specific reduction in IgA in the serum of the mice, but also they have a difficult time responding to particular immune challenges. In addition, we were interested in trying to understand what parts of the immune system are affected. So there are all these different compartments within the immune system that I showed you on that diagram. And so in all three compartments we looked in the bone marrow, in the spleen, and in the intestines, we see a reduction in antibody-producing cells. And you can see here the red bar is low across the board. In addition, we found that there was an expansion of IgM-producing cells in the spleen, so we were able to localize that abnormal increase in IgM production to the spleen of these animals, although we're still trying to investigate exactly why that's happening. We also then looked in the intestines at a very important type of cell in the intestine called the plasma IgA cell. And we found that those are the cells that pump out IgA and protect the gut, and it's one of the most IgA-rich parts of the body. And we found that the plasma cells that make the IgA in the intestine are reduced in the Kabuki mice, and in general they just have fewer B cells in the gut in general. When we looked at the differentiation state of the cells that are there, we found actually there's an expansion of immature plasma cells in the Kabuki animal gut, suggesting that there's a block in differentiation. So they start to form a plasma cell, but somehow they get stuck. And the Kabuki plasma cells aren't quite as good at taking the final steps to become a fully differentiated, mature plasma cell as those in the wild-type animals. Let's see. Sorry, it's not advancing for me. And then in addition, in collaboration with the Bjornson lab, we looked at the number and size of pyre patches that are found in the intestine of the Kabuki mice. And they were found through the elegant work of a very talented graduate student in the Bjornson lab, that the number of pyre patches in the intestine of the mice is lower and that their size is smaller. So this suggests that perhaps there's a developmental defect in the formation of these special structures in the gut where the IgA is made. So in general, the Kabuki mice have lower IgA levels just like the humans do. They have splenomegaly just like the humans do. And they reveal new insights into the reason why this is happening. We believe that there are reduced numbers of IgA producing cells across all the compartments of the humoral immune system, and that this is likely because of the differentiation block, that the B cells have a particular problem taking that last step to become the plasma cells. And as has been suggested by many of the groups that spoke today, we believe that there might be therapeutic interventions that could help the B cells pass through to this final stage of differentiation and improve the antibody production in subjects, in human subjects. And so that's the ultimate long-term goal of the work in my lab, is to utilize models like the Kabuki mouse to try to find new ways to improve the immune function of subjects that have Kabuki syndrome. So in summary, Kabuki syndrome patients, the majority of type 1 Kabuki patients will have some deficiency in IgA production. Many of them will have low memory B cells, and the Kabuki mouse model is a useful model for studying this and trying to come up with new therapies. So I would like to acknowledge the work of my lab, the collaboration with the Bjornson lab, and the support of my sponsors.